okay? And this is enzyme. When your effector, yani molecule, can control the activity of enzyme, when it's substrate, again, it's considered as a homotropic. And this is a positive. Yani what? Yani, for example, I'm a glucose. Okay, I'm a glucose. And I want to describe the glycolysis because I know that you are familiar with glycolysis. Okay, so you should know that glucose considered as a positive effector for the glycolysis pathway. Yani glucose say that, hey, I'm here, increase the rate of reaction. Yani positive effector. Do you understand?
Yes.
So when your substrate is similar to your effector, it is a homotropic effect. This is the simplest way to describe the homotropic. But what about the heterotropic effect? Heterotropic effect yani other molecule. For example, your glucose as a substrate. A molecule that would control your activity, for example, is, I don't know, citrate, malonate. The nature is different from substrate, okay? So the effector means that heterotropic, other type. What is the example? Just know that. Citrate, for example, considered as an effector, again, inshallah, in a metabolism of carbohydrate. Dr. Imam Wali will describe this point. Phosphofructokinase can inhibit. So I say that inhibit. So it is a negative effector or positive? Negative effector actually. And especially for the metabolism, you know, these are the basic concept of metabolism. If you didn't understand this session, you cannot understand metabolism. Okay? So, as I mentioned, substrate is a positive effector. It means that all of the enzyme, be ready, I'm here. Okay, when I accumulate here. But what do you think about end product? You have an accumulation of end product. For example, for glycolysis pyruvate. What do you think about the signal? It says that activate or inhibit. Inhibit. Okay? When you have an accumulation of product, you say that why do you want to waste your energy? Stop. Okay? It means that they send a negative signal. And this negative signal now as a feedback inhibition. Okay? We have a lot of feedback inhibition in a metabolic pathway. In a carbohydrate metabolic pathway, in amino acid, in a lipid metabolism, okay? So you should understand the concept. It's very clear. When substrate module, pathway should be activated. When product module, inhibit. I'm here. What do you want? Waste your energy. Is it clear? This is the definition of feedback inhibition. What about the multiple form of enzyme? You know, as a physician again, because maybe I cannot describe by detail, because as I mentioned, inshallah, in Farsi we say بشرط حياة. Inshallah, for the next semester, بشرط حياة يعني if I be alive.
Yes.
Okay? Inshallah. I will describe this actually form of the enzyme in one session. Yani, I require at least 2 hours to actually explain the isoenzyme and actually the role of this molecule. But just know that. For example, sometimes you have a different form of واحد enzyme, one enzyme. For example, about, I don't know, lactate dehydrogenase. Or I don't know, for example, I mentioned, which example here? lactate dehydrogenase. It's better at first to describe here. This is a definition of the isoenzyme, and it's better to talk about in this slide. You know, lactate dehydrogenase, I know in Iran we had this enzyme in high school. What is the role of this enzyme? John. Yes.
I think I'm not sure, but I think it removes the proton, so the lactate changes into lactic acid.
So, at first, I want to know that. Have you ever heard the anaerobic metabolism?
Yes.
I think all of us experience this situation. We have to run very fast and we feel a cramp and pain in a muscle. I think all of us, for example, for reaching to the exam, I don't know, reaching to the taxi, to the bus, I don't know, everything, okay? Why you have a cramp or pain? Because normally, if you have enough breath, enough oxygen, the end product of the glycolysis is pyruvate. When you run fast, you are at risk of hypoxia. Yani oxygen موجود. Oxygen, when is absent, pyruvate is not end product. Lactate is end product. And lactate is lactic acid. This is acid. Accumulate acid, lactic acid in a muscle. And it can cause a pain. Muscle fatigue. Okay? Fatigue in Arab, what does it mean? Do you understand? Yes. So, what is the name of enzyme catalyzed the reaction? Lactate dehydrogenase. But lactate dehydrogenase, there is no one of this enzyme. We have a different form of this enzyme. Lactate dehydrogenase. For example, lactate dehydrogenase 1, 2, 3, 4, 5. Okay? We have a different form. For example, this is the M. M yani originates from muscle, H originates from heart. Okay? At first, you should know that enzyme is tetramer. By combination of M and H, you have a 1, 2, 3, 4, 5 form of lactate dehydrogenase. For example, H4 found in which organ? Most of them originate from heart. So, for example, lactate dehydrogenase 1. And for example, lactate dehydrogenase 5. And sometimes represent as a LD. Okay? Lactate dehydrogenase 5. Where can I find it? Especially, for example, liver. What about actually number 1? Heart and especially RBCs. Okay? So, as a physician, what is the importance? Because I want to talk about the last part of my lecture, I mean the serum enzymes, better to describe here. You know, as you can see here, we have a different form of enzyme. Different form of enzyme known as a isoenzyme. Isoenzyme number 1, isoenzyme number 2, and sometimes isozyme abbreviation. So, what is the characteristic at first? Maybe they have a different amino acid sequence. What about the key point of an isoenzyme is a different compartmentalization. Yes, they have a different, again, velocity. Yes, they have a different Km, different Vmax. Okay, what about actually the practical point as a physician? You should know that they have a different distribution. Okay, some part in liver, some part in an RBC. So, what is important? What do you think about the? I want to know that. What do you think? So, how can I use from isoenzyme as a physician, inshallah, for future?
For describing drugs. For describing drugs. Like when you know when it's work and how it works. You will describe the right one for the right side. For like.
For example, I want to ask a question because I want to know you understand or not. For example, as a physician, you read the lab report, okay, and you see that, lactate dehydrogenase 5 is abnormally very high activity. What does it mean?
I should describe. So by using the lactic dehydrogenase, we can use it to regulate the work of the enzymes and lower the rate or increase according to what needed in the patient.
No, it's not related to the regulation strategy. It's some clinical point. You know, when lactate dehydrogenase 5, because يعني, for example, this is a hepatocyte. Hepatocyte يعني liver cells. I know that lactate dehydrogenase 5 is present in a hepatocyte. Appropriate site in a hepatocyte, not in a bloodstream. When they enter to the bloodstream, what does it mean? Why? Absolutely, there is a damage here. Like a hepatitis, like a consumption of alcohol. When you take alcohol, your hepatocyte became damaged. They cause a breakdown of cell membrane, they enter to the bloodstream. This is the concept of serum enzyme and isoenzyme. For example, please tell me, especially as I know, lactate dehydrogenase 1, this is the RBC. Lactate dehydrogenase number 1 can frequently found inside the RBC. Please tell me, when you observe a lot of this enzyme in the blood, what happened? Leak from of what? RBC. What happened? And when you have a leak from RBC, you know, rupture. When you have one specific disorder, you know.
Hypertension?
No.
It's transport.
Lack of hemoglobin. What was the name of the lack of hemoglobin disorder? Anemia. Okay? Inshallah, again in a vitamin session, you will be familiar. We have a different type of anemia. When your RBC lysis, this is known as a hemolytic anemia. Okay? Yani hemolytic anemia, yani your RBC breakdown, release any content, release hemoglobin, release lactate dehydrogenase 1. Is it clear? This is the concept of the actually isoenzyme. About the details is not important, okay? Just understand the concept. About the covalent, about 10 slides are remaining. If you agree, I can continue, okay? Because maybe 15 minutes, okay? So, about the covalent modification, actually covalent, I'm sure you're familiar with this covalent, but maybe you didn't actually remember. This is the enzyme, okay? As I mentioned, sometimes you add phosphor to your enzyme for activation, okay? Because this adding of the phosphor is through covalent bond, this process known as a covalent modification. Is it clear? This is the concept of this slide. Covalent modification, yani following covalent bond of phosphorus, for example, sometimes you activate your molecule, sometimes you inhibit. But again, you should actually memorize this point for the next semester. It's full of points, inshallah, for the next semester. I cannot say that all the time, if I add a phosphor to my enzyme, I activate my enzyme. Sometimes after adding phosphor to your enzyme, you inhibit the activity. Sometimes, for example, if you want to activate this enzyme, what do? What should you do? You should remove the enzyme. Phosphorylation, dephosphorylation. Okay? This is the name of process. Phosphorylation most of the time mediated by which enzyme? Kinase. Okay? And dephosphorylation most of the time catalyzed by phosphatase. So kinase, phosphatase. Okay? And most of the time, actually, for regulation, the source of the phosphor is ATP. And know that, you should know that. For example, maybe I ask you some true and false, especially these two lines is important for me. Okay, in this slide. For example, if you add a phosphor to this enzyme, glycogen phosphorylase, you increase its activity. In contrast, if you add a phosphor to glycogen synthase, you decrease the activity. Do you understand? So we cannot say that there is a fixed rule. If you add a phosphor activate, depend on your enzyme actually, you can activate. And other molecule you can add, this is the acetyl group. In addition, for example, phosphor, sometimes you add an acetyl, sometimes it activates enzyme again, sometimes it inhibits the enzyme, okay? This slide is for more information because I want just to talk about this one, as I mentioned. Enzyme in the presence of ATP became phosphorylated. And as I mentioned, sometimes it is active, sometimes it is inactive actually. But the remaining is not important. About the proteolytic activation, would you please hold your question because
Yes.
Yes.
Like on the previous slide, I think there's a part you didn't explain about histones.
About histones, this is for more information, okay? This is for more information because you mentioned, I mentioned that we have actually covalent modification. Most of them, because in metabolism, I'm sure, I think, because I want to have fast review about the metabolism of all molecule, lipids, carbohydrates, you don't have acetylation, okay? But you just know that addition of, for example, acetyl is one of the example of covalent modification, and this is more common for histone. Have you ever heard of histone? The proteins around actually of the DNA, the chromosome. The forming of the
The
chromosome.
basic unit of or
Yes. Are you familiar?
in the hospital.
DNA wound around the histone and form a chromosome. Okay? And whenever you have a very active translate gene, for example, some genes translate for 1 million times. When they want to translate for a long time, the histone, this is for example, histone molecule protein around the DNA. Okay? For example, this is the DNA. It means that here you have the acetyl group and the genes that are near here are very active. They transcribe for the several times, okay? This is the concept of this slide. The acetylation of the histone. This is the controlling of gene expression because I'm sure Dr. Panahi, inshallah, will explain. I didn't want to focus by the text. And this is for more information. Have you ever heard the name of trypsinogen and trypsin? Pepsinogen, pepsin. No? What do you think? Pepsinogen, pepsin. You don't have any background information?
Pepsin is the enzyme of
Both
digestion.
of them are enzyme.
Of the digestion.
Make Pepsi.
Making
Pepsi.
pepsin. What is the precursor? Gene يعني precursor. يعني inactive. Okay? So, another strategy to control the activity of enzyme is a proteolytic cleavage. يعني especially in a gastrointestinal tract, like stomach and pancreas, you have a zymogen. Gene يعني inactive form. Okay? Like, for example, trypsinogen is inactive.